So my name is John Shepard. I'm a professor here at the University of Hawaii Cancer Center, and I head up the Artificial Intelligence Precision Health Institute and the Affinity group that sponsors these talks. This is where we hear from leaders of the field, discuss trends in AI and healthcare, bring together researchers from really a lot of different areas, as well as clinicians and advocates, with the goal of fostering interactions to solve problems. The format is roughly 30 to 40 minutes with interactions afterwards. Like I said, I love it when people leave their cameras on so we can see each other and interact. And if you have questions or feedback on suggestions on speakers, please let me know, or Peter Washington. Is Peter on, by the way? Don't know if he's going to be able to make it this time. Okay, so this talk series is sponsored also by the Hawaii Data Science Institute, which is a major collaboration that trains students to do AI and machine learning, and also the University of Hawaii Cancer Center. And this is our facility in the Kaka'ako campus right outside of Waikiki. We do a variety of projects and have openings for graduate students as well as postdocs fairly continuously, and always welcome people to consider joining us. And you can go to our website to learn more about us. We have very generous compensation for speakers, which amounts to a beer stein with our logo on it. So just to disclose, all speakers are being generously compensated for their time. And the reason I say this, it's actually funny. I would ask people to do it nobody wants to do Zoom calls anymore. And even if we paid people a couple $100, nobody wanted to do it. But a beer stein, people go, okay, a beer stein. So that's worked out well for us to get some really high quality speakers, just like today's with Dr. Zhang. So Dr. Zhang is a researcher in cancer biology at the University of Hawaii Cancer Center. He has over 20 years experience in industry, government, and academia, primarily doing drug discovery and development. And his research encompasses many areas, including multi-omic data, implementation of novel computational and intelligent algorithm models, and more things that I can really list here. He joined the Cancer Center only about a year ago, and he's published numerous high-impact articles in Science, Nature, and so forth. So we've asked him today to give us a update on what he's doing on AI drug discovery and development, and I'll turn it over to you, Dr. Zhang.
Well, thank you very much, Dr. Shepard. Good morning, everyone. It's my great pleasure to give you this presentation today, and happy Friday. So I really like the cup. It really looks really nice. It's a good composition. Probably we should have some beer together with that. Yeah, so let me share my screen, and so just sort of like a point out a correction. It's not really a correction, but I have been here for 4 months. Not over a year.
Oh, seems like a year. Sorry.
Can you see my screen now?
We see the presenter view.
Okay. All right. Can you see it right now? Okay. All right. That's great. Thank you. Sorry about it. So as Dr. Shepard just mentioned about it, in my lab we have to do a lot of things like different build, different targets, including both the software development, algorithm design, as well as the medicinal chemistry, high throughput screening, and collaboration with clinicians for clinical trials. So, but today I will really focus on since the audience here mostly computer scientists and AI developers, I will focus on the AI-based drug discovery and development. Talk about what we have done, what's the success, and what's the potential challenges in the future. As many of you may know, drug discovery and development is probably one of the most complex problems to solve. The key is that from a huge, gigantic database. What is this? Why there is this line here? I don't know. What is it? It's on me or from somewhere?
Amy, I noticed that there's a blue lines that are being drawn by, maybe you can look into that. It's not me and it's not him.
Yeah, right. Yeah, but that's okay. But anyway, so
We'll investigate. Go ahead.
yeah. So it's the key problem is that you have to identify sometimes one or few compounds from a gigantic database. I mean, how big is a gigantic database? I always give the analogy of our universe, right? So when I was little, when I would look into the sky, I would say during night, I would say, oh, how many stars? We have so many stars, countless. How many stars in the sky? I mean, anybody has any idea how many stars do we have in the sky? So, I mean, somebody did an estimation. It says that we have about 1 billion galaxies in our universe. And in each universe, galaxies, we have, there are about 1 billion stars. So, which means it's totally is about 10 to 24th power, right? So it's billions of billions. But considering the small molecule chemical space, we have a big number, like it's 10 to 60. So compared to our universe, the number of stars in the sky, this one is much, much larger. And in the biological space, like how many proteins theoretically we can design based on the sequence, right? In our body, we only have about 200 million proteins working. But theoretically, this number is really, really huge. Is 160 zeros behind one. So compared to our astronomical universe, all of these protein, number of proteins, number of small molecules that can be theoretically designed and synthesized, it's really complex. And as I mentioned that, when you identify one of them, you have to consider that, they have to good efficacy, they have good bioavailability, they have to be safe, right? So it's a really multi-parameter optimization problem. And really for a system, actually, we don't really know too much about the human physiology, when it's in patient and the pathophysiology. So we don't really know too much. So we have to deal with all of these different problems. This is the pipeline. We call it the pipeline of drug discovery and development. So really from the disease mechanism, from the target identification, validation to hit lead discovery and optimization, IND enabling studies, clinical trials, and FDA approvals and marketing. So on average, it takes about 10 to 15 years and $10 billion to bring a new drug onto the market. So it's a really costly and time-consuming process. That's why every technology, any methods that can speed up this process will be highly appreciated. That's why computers have been really applied in almost every stage of this pipeline. For us, we are more focused on this stage, sort of more like the predict protein structures to look at the properties, toxicities of the drugs, and to do chemically rational design of the molecules, to understand their enemy tox in the body and the interaction with the body or the body interaction with these drugs, right? So we'll focus on this part. And of course, we also do some genomics, bioinformatics analysis studies, but more focus on the small molecules. Sometimes we call ourselves as chemoinformatics instead of bioinformatics or genomics. Just a little bit about the history of AI. So before going further, here is the riddle. Anybody knows about these words about? Well, basically, this represents the 7, we call it the Magnificent 7 AI companies. So the 3 A's means Apple, Alphabet, and Amazon, and Meta, Microsoft. Of course, we will have NVIDIA. If you buy NVIDIA stocks, you must be rich now. And we have OpenAI. So that's if we arguably you can put I there, that means the IBM and Intel. But of course, by looking at their stocks on the market, they are in big trouble. But they have developed AI-based technologies, hardware and software as well. As I said, it's a little bit about history. So AI development really started in 1940s, 50s. And then at that time, the computational power was not enough. The technology was not enough. So people found, the idea was exciting, but it was not really helping the advance of the technology and the science. And in 1970s, basically we call it the winter of AI, right? But in the later 90s, in 1997, the IBM developed this gigantic big computers, Deep Blue, and beats the international chess champion Kasparov from Russia. That was a big milestone for AI. It's like a machine can really beat human in the gameplay, right? And then after 20 years, in 2016, another company, it's a UK company, DeepMind. At that time, it was a small company, AI-based. They developed this new AI program called AlphaGo here. Actually, instead of doing the international chess, which is a 9 times 9 grid, right? So the chess, the Go chess actually is 19 times 19 grid. So previously it was intractable. So it was computationally too expensive to do this. But now with this new technology, actually it beats the world champion in South Korea for the Go chess. And the same company later was acquired by Alphabet, the Google at that time, right? So I think based on AlphaGo, they developed this AlphaFold. They published the paper in 2021, and early this year they got a Nobel Prize. Everybody knows about it. But by now, it's sort of like by 2021, still, all of the AI is still in the expertise area, right? So it's in the specific computation or chess or protein structure prediction. It really solved we call it the 100 years of problem in biology. That's why they got this Nobel Prize, although it's in chemistry. I thought it was really interesting. By the end of 2022, just 1 year later after AlphaGo, in November 2022, then OpenAI came out, released the ChatGPT, but within 1 month, they collected like 1 billion users. So now, from the expertise area, really becomes a general public interest, right? So everybody now can play ChatGPT, can use AI. Everybody was talking about AI right now. That's why we give this talk today. But as a reality is that, we work in the area, we always follow the development. The AlphaFold, although it was published in 2021, but we knew, as people in the field, we knew about this actually in 2019. So there is this interesting comment from Bloomberg. It says that, actually the tech company actually started doing drug discoveries because the drug companies actually, they are doing really bad jobs. The failure rate usually is 95% or sometimes 99% of failure rate, right? So with big techs, actually, they can come into the field, and with their technology, with their capital, they can do a lot of things. Usually in my class, when I teach the students, I usually tell them, this is the way you need to think out of the box. In the current state of the world, in technology, in science, there is no really clear line between different disciplines, different sectors. So, as I said, the tech company now can do drug discoveries. The other example I usually give is about the digital camera, right? So if you think about it, 10 to 15 years ago, you took pictures with these films, right? At that time, Fuji films, Kodak films, they are the competitors, but they are not the people to put their competitors out of business. It was a different thing. It was from Samsung, from Crucial Technologies. They developed this digital, the digital memory sticks. Those digital memory sticks actually put the business, put the films out of business, right? Unless you are really hardcore professional photographer, probably we may still use them. But today, how many people are using these films to take photos, right? The same thing for phones. Now the phones will be used as digital cameras to shoot and to point and shoot instead of use digital cameras. So in different areas, the disruptive technology may become very different areas. It sounds like an irrelevant field from one discipline to another. This is the landscape of the AI drug discovery right now. As you can see here, this is traditional pharmaceutical companies, and this is the AI specific, they use AI as startups, and then these are the investment company, venture capitalists, and these are big tech companies. I can envision that in the future, these 4 circles will come closer and closer, overlap more and more, right? So there is no really clear line between them. You can always do something, develop disruptive technology from one field to apply to another field. These are the AI specific startups for drug discovery and development, and along with their pipelines. It has been a lot of questions about whether AI really helps drug discovery and development. And indeed that, there are a lot of pipelines going on right now. They are really promising, but in reality is that there are a lot of setbacks during the last, I would say 3 or 4 years. But as I said, drug discovery and development usually take 10 years. We still need to have more time to take a look at it. But indeed, like Accenture, they had first AI designed therapeutic agent in clinical trials in 2020. And 2 years later, in October 2022, they discontinued the program. Of course, what they are saying that is not because of the AI program, it's because of the switch of their strategic portfolios. Recurring, which they promised within 10 years, first 10 years, they will provide 100 clinical drugs, but now more than 10 years later, they only have 4 clinical candidates in the clinical trial. So there are a lot of hiccups and setups, but we still need more time to look at it. So that's basically the business, the landscape. When we talk to investors, venture capitalists, we have to tell them, what are the competitors out there. So for ours, we have been interested in traditionally, during last 15, 20 years, traditional computational modeling, like try to develop machine learning programs to study the drug activity relationships, and also develop algorithms to study how the drugs interact with their targets structurally in 3D. And from this, we can identify hits, optimize lead compounds to get the more effective therapeutic agent with less toxicities. And recently, we really want to, as Dr. Shepard mentioned, we really want to integrate the big data, multi-omics, genomics, bioinformatics, transcriptomics, metabolomics, all of these data together with the latest stage of the AI technologies to develop, this integrated approach to study precision medicines, personalized therapeutics, particularly in cancer. We even develop a technology for molecular viralization, use the Meta hardware, the Quest, as well as the Microsoft HoloLens for this kind of virtual reality and augmented reality viralization for small molecules. Literally right now, you can sort of like walk into the molecules and discuss where is the important part to target. Another area I'm very interested in is we call it drug polypharmacology. As some of you may know, when you develop a drug, no matter how well it was designed, how specific it was designed, when they get into the body, they always hit multiple things in our body, interfere with the multiple pathways. So on one side, this will cause we call it off-target toxicities. But on the other side, they can be used for different indications, right? So one drug like Gleevec, it was the first kinase inhibitors designed for leukemia targeting BCR-APRIL, but later on they found they target multiple kinases in the body. They can be used for GI cancers, some other cancers as well. So the idea is that we can integrate all of these traditional and the latest, the cutting edge technology together to build our unique platform, more robust, unified platform for drug discovery and development. On the other side, as I said, we also do work with clinicians, biologists, chemists to do the develop therapeutics for different targets. So, primarily we focus on 2 pathways, PR3K, AKT pathways and the Perils pathways. But as I mentioned, in our bodies, all of these pathways are intertwined together. So one of the group of proteins we are interested in is called the plex-strong homology domain proteins. All of these proteins, they have the same domain, 100 to 120 residues. They have the same 3D fold. Basically, it's 1 alpha helix followed by 7 beta sheet. There are about 300 of these kind of proteins in our body. So they are very critical for signaling, for molecular translocation, and for cancer metastasis, development, as well as the cell skeletons, the structure, the matrix of cells. Another area we're interested in is we call it protein ubiquitination and degradation is the the process in our body to maintain the protein levels in our body to make it not too much, not too low, so that if too much, it can cause the cancer. If too low, it can also cause cancer or other diseases. I mean, all of these are novel biology, novel targets, no drug developed for them, and that's why it's new, it's interesting. But on the other side, as I said, we are interested in drug repurposing. So there are some well-established targets like EGFR. There have been 3 generations of drug developed for it, but they keep get a resistance, right? So some of them, in some patients, actually within 3 months, they can develop resistance. So when this happens, we have to target them resistance to develop new therapeutics to overcome the resistance. So some other technologies, other targets, instead of proteins, we are also interested to target the RNAs. Some of you may have known that early this year, the Nobel Prize in Physiology is for the microRNA discovery. Victor Ambros identified the first letter 7 microRNAs as they found that these microRNAs, they call it non-coding RNAs. Previously, they would think that they were junks in our body, but later on, they found that these microRNAs, non-coding RNAs, actually they are critical for disease development, signaling, as well as genetic, genomic regulations of the cells, of the growth. So we are trying to integrate, target these RNAs for drug discovery and development as well. So because of the audience is more about the computational, so I will focus on the method development. I just found that I only have 30 to 40 minutes, so I will try to be as fast as I can. I have 60 slides. So the idea of this is that we develop something that we can screen, all of the, if not all of the cancer cell lines, most of cancer cell lines that's ever available in our body, in human, right? So the real story behind this is that when we develop these therapeutics drugs targeting the E3 ligase, the ubiquitination and degradation, we talked to the venture capitalists because our agent was primarily focused on the breast cancer and the prostate cancer, and of course there are a lot of competitors in this field. So the venture capitalists asked me, say, do you have any data? I mean, the data really promising. The target is interesting, but do you have any data in rare cancers? So because in rare cancer, if you develop therapeutics, the approval will be sort of like faster. They call it the breakthrough designation, right? So usually the investors like this kind of work. It's novel, and it's also the regulatory approval will be faster. So I said, no, but we can do a lot of tests. So when I came back, I said, it will be difficult to find collaborators for rare cancer, because for rare cancer, usually there are only few people working on it, right? But NCI, there is this established, they call it NCI 60 cell lines panels. So I said, why don't we do that? And plus, it's free. So you just send them the compound, and they do the test, give you the result back. But of course, you have to deal with these material transfer agreement, and et cetera, et cetera. At the beginning, I give them 5 compounds to test all of the 60 cell lines. I was expecting maybe within 1 month, 2 months, I will get the result back. But it turned out at the end, it took them 6 months to give me back. I couldn't complain because it was free, right? Or what can you expect with a free service? But this gave me some thinking, some thoughts. Say, okay, can we develop something that can be faster instead of only 60 cell lines? Can we, screen all of the possible cancer cell lines that have been developed or manufactured, right? I mean, not only breast cancer, prostate cancer for these common cancer, but also for rare cancer like hepato carcinoma or the sarcoma. They only have few cases in the world. Not a whole lot of people to do that. There was no data, no model for that. So that was basically the idea, is that we want to develop this AI platform with the big data. Maybe with the transfer learning, with at that time, this was before COVID. There was no large language model. I mean, there was scientifically, but not like ChatGPT came in. After ChatGPT came out, right now we have the chemical language model, we have genomics language model, we have a protein language model. different things, right? We can use these different technology together. But at that time, we want to develop something new algorithms about, instead of the plain convolutional neural network, we can look at the, new technologies. So this is the big, general the procedures. We curate data, we split them, we do the training, and then we select models for them. This is just general. So the first one is data curation. Right? As I said, data is the foundation of machine learning, of AI. And we focus on some big data sets. So one of them is called European curated called Campbell. Basically it's everybody in the world. When they did the screening of chemical compound for activities, they can deposit the data there. So you can expect that the standard is sort of relatively low, and the data its quality is low, and a lot of noise and a lot of wrong data. So we started from over 3 million data points for 300,000 compounds covering 1,200 cancer cell lines. So at the end, after filtering all of the wrong data, the noisy data, everything, we got 1.5 million data points for 140,000 compounds. It's less than half of them, right? So also we did this for the GDSC. The reason I'm talking about the procedures are very similar, but in this slide I want to point out is that, as I mentioned, Campbell is a worldwide effort. Everybody can deposit their data, screening data. But the GDSC is NCI funded and coordinated efforts. So they have very rigorous standard to deposit data. But even with this national coordination and the standard, rigorous standard, and we found that the data actually is also dirty. So for example, they claim that there are about 520 compounds, drugs screened. And we found that actually only 440 of them are unique. Just because some screening groups, they just have duplicates. Some, for example, Gleevec, the brand name is Gleevec, right? The generic name is imatinib. So some of them, they use different names, and they don't have expertise in the area. They thought it was different drugs. So they put the data there. It was annotated as different drugs with the different screening. So as you can see, even with this kind of rigorous standard, you can still see that the drug is, the data is dirty. That's why the data curation in AI, in machine learning, is a big challenge. And even some of them, actually, this was screened by Sanger. And you can see that there is no information about these drugs. They don't have structure. Nobody knows what they are. They just put this, we screen this compound, and we got this activity. We even contact Sanger, that nobody knows about it. And we have to discard them because, the big problem, I think, in the data science in this area is that people just copy paste of this data. We cross-check multiple databases, multiple resources from multiple publications. They just say, we got this compound tested, screened, and we got this. It's from multiple data, they just copy the same thing without any adding information. So, for us, the structure, the name, the information of a drug is critical. So we have to discard them. So at the end, from this 518, we only got 400 unique drugs. And basically, with this national effort, 20% of the data actually are not really usable. So I think that's one of the challenges in this area. This is the overall. So I don't, because of the time, I don't want to go into details, but starting from this 3 data set, at the end, we got about 1.8 million data points for 140,000 compounds covering almost all cancer cell lines, 1,500 cell lines. And then we need to, that's the chemistry data, biological data, right? Now we have to go to the genomics data and the bioinformatics data for cell lines. So after considering that, it's further reduced, basically, almost half of the cell lines, they don't really have genomics data. So at the end, we want to look at both the expression, for example, mutation data. So now we have the largest data we have is about 130,000 compounds. Now for mutation, it's 1.6, but for both of them, both the mutation and expression data is only 1.4 million data points. So now we have the data, how to represent them, right? So as I said, we did this before the COVID. There was the large language model was not that popular at that time. So we know that AI was good at image processing and recognition. So we try to convert everything, the chemistry, into image to do the process. But previously when we characterize a compound, we use different ways. We use fingerprints, we use a molecular descriptor. It's sort of like when you describe a person, you say, okay, now what's the weight of this person? What's the height of this person? Where does this person come from? Et cetera, et cetera. The same thing for molecules. We can say, okay, how many carbons in the molecule? What's the molecular weight of these molecules? What's the volume of these molecules, right? So how big is these molecules? We call it molecular descriptors. And the fingerprints is sort of like here, does this molecule has aromatic range, is this hydrophobic or hydrophilic, right? Whether it has a carboxylic acid group. If you have the motif there, it's 1. If you don't have it, it's 0, right? It's 1, 0, 1, 0. We call it fingerprints. And also, these molecular structures, you can think of them as the connections of different dots with edges, right? This can be treated as a graph. We have graph neural networks. And now we know that we have language models. So all of these actually, you can use a string to represent that. We call it chemical language models. This is particularly good probably for natural product, which is unique for Hawaii. We have a lot of natural product resource because they are usually much bigger. For drug-like molecule like this one, molecular weight is about 100, 200. Many other drug-like molecules must be less than 500. Natural product like antibiotics, they can be more than 1,000, 2,000. So they are much bigger. So maybe the chemical language is more appropriate for that. So we have an initiative for natural product-based drug discovery and development. Now, you can have that, use this one. You can not only predict its activities, but also you can generate new strains and new languages. This refers to new structure, right? So we call it generative AI for drug discovery and development. On the other hand, so for genomics data, for cell lines, we develop this called bioimages. We also convert everything into images to represent them for AI software, neural networks processing. But of course, you can right now use genomics language, you can protein language models to deal with that, right? So there are different ways to represent. And this the building blocks. We call it octave residual neural networks. Of course, we can use large language models. We can use transformers. Transformers use the plain convolutional neural network, right? We can also integrate our octave ORNN into the transformer to do these kind of predictions. So the input will be the chemistry, the chemical structures, the biologies, the genomics, and the output will be we call it RC50, which means the concentration of the drug to cure 50% of the cancers. So we can predict this. We can tell that, okay, it's exactly at this concentration, you can really cure 50% of the cancer cells. And this is the real the algorithm behind it. And the ORNN basically is look at both the high frequency and low frequency studies to identify the critical features in the structure to say that, okay, these features are really critical for the activities. That's where we can talk to the biologists, talk to the chemists, to synthesize them, to let them to know how to design new molecules with more efficacies, low toxicities. So this is the mathematics behind it. So I'm not a mathematician, but this is how we do upsampling to convert the low frequency images to high frequency features. And these are our cost functions. This is the way of how we build the model. By the way, so if any of you have any questions, just ask. So we have the representations, as I said, different ways of representations: fingerprints, molecular descriptors, graphs, images, language models, language strings, and then we split them into training and tested, multiple, randomization, shuffling, careful partitions, cross validations, etc., etc. So we of course we always do the ultimate validations experimentally. So this is the, when you have a model, as we always do, we have to do the assessment, right? So you can see that this is the vanilla, the plain convolutional neural network. The convergence is not good, and it's, the loss function is so high. And with ORN, actually, it converges really, really fast, and the loss function really optimizes really, really well. We also look at the capability of our models to handle both the big data and the small data, because some of the data in drug discovery, we just don't have enough data. Like a CACO2, we only have a few hundred compounds. Some of them we only have 10, 20, or 50 compounds. So we can see that it really, CACO2 means it's the permeability, measuring permeability, how the drug can enter the cells, because if it's effective, it has to enter the cell into our body, right? So that's about a CACO2. So we saw that our model can really deal with both large data set and small data set. The prediction usually is about 0.8% with the correlation coefficient. The other side, as I said, why we use RNA, because it's one part of the found this group from Meta, Facebook at that time. So they developed this RNA algorithm initially. So for us, we have to make sure that they are explainable, right? So because we have to understand the biology and the chemistry, ultimately talk to the chemist to synthesize some of the compound. So we developed this, we call it a PAM, prediction activation map, to really project this, the weight and all of the lattice space and back to the molecular structure. Say, okay, this is the point where it can be modified. They are crucial for the prediction of the activities. So, for similar compound analogs, from the initial structures, we can do the projections, and we identify, all of them. These areas are crucial for the activity, the wider range of the activity, whether they are active or inactive. And even for different scaffold, right? These compounds, they are completely different. We call it cross structures, and we could do the same thing, be either for analogs or the same series or for diverse chemical structures. We can do both. And also, as I said, we developed this and applied to one of our E3 ligase project, targeting the ubiquitination degradations. And we work with our collaborators, Dr. Nat Commun and Dr. Jason Lee. And basically, we build our model, we design some compound, we make predictions. We thought, okay, the prediction shows they are really active. So we give them to Dr. Lee and Dr. Winu, and their group will test them in cells and in animals for us. And we did indeed found very active, much active than the original hit and lead compound. Now they are in the process of the further studies in the preclinical animal models, PDX model studies as well. Of course, we also docked this molecule into the active side of the target. We skip to its E3 ligase, and it really fit into the pocket very well. I think maybe my time is limited, so I really want to tell you a little bit about the drug combination, but I think maybe I don't have enough time, so I will skip this one. Instead, I will focus on the application, give you one or two examples to demonstrate, how we use this kind of technology, develop new therapeutics, going through the clinical trials. So this first project was targeting EGFR, as I mentioned. So Dr. John Hemek, he was the chair of the head and neck and thoracic medical oncology department, and Zhitai was my graduate student. He was the driving force of this project. This the lady was our first patient using as a compassionate use of these therapeutics we developed. So briefly about the biology is that, one day Dr. Hemet came to me and said, oh, we have these patients. They have these called exon 20 insertion mutations. So with these mutations, the disease becomes really aggressive. They usually only have like 2 to 5 months of survival. So he said, since you are doing computational drug discovery, can you give us something very quickly, within 1 or 2 months? I was joking, so give me $10 billion, I will do something for you. Of course, we cannot wait. There were no $10 billion, but I say, we can look at we cannot develop new drugs on new targets, right? That will take forever, 10 to 15 years. So, but we can look at all of the approved drugs. So we have about 1,500 approved drugs approved by FDA. So we can just look at those. Since we have intensive studies on their toxicities, we don't need to worry about that. If they show efficacies, you can directly use them in clinical trials. So that's what we did. When we have this real project, we don't only use one method. We just use any method available at our hand, right? So we develop this drug target, graphs. We can, as I mentioned, we can do high throughput docking to dock each of the compound drug into a target EGFR and look at the fitting, which the drug fits perfectly. We evaluate chemical similarity, genomic similarity. We develop these AI programs to predict the IC50s. We combine all of these methods together. We give them 11 compounds. Now, these are the 3 generations of the EGFR inhibitors. The first generation, alutinib, basically for the wild type. The affinity is for the T79050, 95 mutations. And osimertinib was first generation. At that time, it was just approved for the covalent bond inhibitors targeting the 797 mutations. And we found that through our modeling, we found that poziotinib actually was perfect for these exon 20 insertion mutations. It fits the target really well. And it was a shelved drug because of the toxicity. It was first developed by this company from South Korea, Hemi Pharmaceuticals. But they discontinued the clinical trial because of toxicities. But we found that it was perfectly fixing the pocket of EGFR exon 20 mutant. This is the comparison. The gray is the wild type, the cyan, and the green was the insertion. As you can see, the pink is the insertion, right? So with the insertion, there is this shift of this alpha helix, and also this, we call it activation loop here, downstream, down forward shift. So this osimertinib in 797 mutations without the insertion. So the pocket is big, it fits very, very well. And this is with the insertion, basically the pocket becomes smaller. The osimertinib cannot fit anymore, but poziotinib, the new drug we predicted, actually fits very well. Later on, they actually developed solved the crystal structure. There is this back pocket there, and it really confirmed our prediction. I was really surprised and happy that our modeling was so accurate. So they quickly developed cell models, test them, and indeed, poziotinib shows like 67-fold more selectivity than all of the other EGFR, previous generation of EGFR inhibitors. And they also developed animal models, GEM models. They saw that after 3 weeks of treatment with the poziotinib, you can see that the tumor burdens, the growth of the tumor significantly reduced. So, and then we have this compassionate use, as I mentioned, this patient, right? She was dying, so she would like to use any drug that can save her life. So you see within a month of treatment, the significant reduction of the tumor and the tumor burden, and as well as the metastasized remote lymph node, right? The lesions. And even in the blood, there was no detection of these biomarkers for them. So we were so excited that we started the phase 2 clinical trials, we plan to recruit more than 100 patients it was the largest clinical trial for this subset of patient population, actually. By the end of 2017, actually, we finished 11 patients, and you can see that 8 of them showed a significant reduction, 30% of them with the response, and even 3 of them they didn't have more than 30% response, but they also had some partial response. And these patients were previously treated either by chemotherapy. These 3 actually were treated by some claimed exon 20 insertion inhibitors by other companies. They didn't have any response there, but at least there was some response here. And also the CT scan shows the more than 50% of reduction of the tumor volumes. So these are the clinical trials, and the drug actually was designated as the breakthrough for FDA approval. Of course, instead of EGFR, we also targeted, repurposed the other therapeutics like here, a hedgehog inhibitor, vismodegib, which was primarily used in basal cell carcinoma, BCC, but we repurposed it to treat the lung cancer. So this was another success story. We have several others. So how much time do I have, Dr. Shepard?
You're coming close to the end. I would say about 3 minutes to give people some time for questions.
Okay, I will be quick. So basically, the last one is not really about cancer. It's instead about HIV parties, HIV patients. So the reason I want to talk about is that it's one side is for drug discovery is the novel biology, novel mechanism, novel targets, right? So on the other side is that to build models, right? So to be really accurate. I think that's the problem in machine learning, in modeling. So here, we build a model, actually, we can predict almost, I wouldn't say 100%. It's in our cross-validation, we always achieve 98, 97, 98% accuracies. So that's why I want to mention about this, but, I'll be very quick. So basically, these statistics, by to date, more than 100 million, 200 million people got infected by HIV and, across the world, and 30 million of people died of HIV. And if you look at data, actually, they have will be increased resistance in different countries, in different part of the world. And that's why we really need to address the resistance. I mean, there are many resistance mechanisms. One of them, is the HIV protease. I have been long interested in this target when I was in graduate schools. And basically, HIV protease is sort of like a Caesar, right? It need to cleave these proteins produced by the virus. They will become functional, and the virus become matured for infection of the people, of the patients. But if we develop a drug here, it will bind into the pocket of the HIV protease, and the Caesar will not work anymore, cannot cut the proteins, and the virus cannot mature, so will not be functional. But if there are mutations here, the drug cannot bind and will become resistance, and the virus will become active again and become infectious again. So that's the mechanism of the HIV protease. Amazing things that about nature, I'm surprised is that for this HIV protease, it's we call it homodimers. Basically, it has two things that come together from this shape here, and it's almost every residue of this drug in this sequence, primary sequence, it can be mutated. The largest one, it has 99 residues in each monomer, and the largest one I have seen is that 87 of them can mutate it, and it's still, the protein still function. So this is how virus, the nature can evolve to escape from the treatment. So it's really amazing to look at this kind of biology. So of course, as I mentioned, the data is that we have really accurate data of these things, of HIV protease, the HIV from patients. They have data with some of them, they either resistant to the treatment or they are still susceptible to treatment. And it's very specific almost on every residues of them. And this is the way the workflow we have been doing. So we have these collections, we fine-tune. It was based on BERT, large language model. This group from Hebrew University, they developed the protein language model. They call it protein BERT. Basically, you can tokenize the sequences and do protein modeling. So this is what we did. We fine-tuned the protein BERT models and try to predict the resistance to each of these drugs with the different mutations. And this is sort of like the result of them. You can see that as the raw curve shows here, it's really almost, if you look at the curve, it's almost 100%, right? I mean, 98% of them. And this is first to show the accuracy and precision of the recall for both the training set and test set in our study. We did this multiple times with different ways of split. And you can see that always about 97, 98, 100% accuracy and precision. That's why I really want to mention about this. I mean, this is also sort of like we analyze, for some of them still there are some outliers. Why the outliers, or which of the sequence, why they have. But it turns out that when we have these outliers, we couldn't predict them well. It's because of these mutations. They call it accessory mutations or minor mutations. They don't have a lot of data in our database. That's why the model cannot really predict them really well. One of them actually is really interesting is that we predict it as a negative, but it is resistance because it's close to the binding site. This is the IDV, the Indeneria, the drug treatment, and it's close here. That's why probably it affects the binding. So with that, I think just a basic summary of them. So I mean, somebody will really like the AI, like Elon Musk is a supporter of AI, but when he exited from OpenAI, he said, "Oh, we need to stop for 6 months, at least 6 months for AI development," and he started his own XAI company, right? And now, several days, I think early this week, I saw that he got a priority to get the new generation GB200 NVIDIA GPUs spend over $1 billion to build the largest AI clusters in the world. So really powerful. Of course, Yang Leifeng, he's the chief AI scientist at Meta. He's sort of like the opposite. He thinks AI is still in early stage. I mean, for me, I sort of like agree with both. So I think it's still in the early stage. We still don't know too much about it. Still a lot of things to do. And I think at the high level, we have to have a regulatory involvement. Like, there is this case in the High Court. This guy developed an AI program. With his AI program, he developed some product and he claimed the patent, right? And the video is the undersecretary of the USPTO, and his patent application was denied. Now they are still in the court fighting. So it's sort of like, if you have an AI development, what's the patentability? Does the patent belong to whom? So if you are the person developing AI and the AI produce the product, what does the product belong to? Belong to the AI or belong to the person developing the AI? Right? So we don't really have clear rules or laws on this. And if we develop AI, a drug with the AI, will FDA approve it? Right? So we need an involvement of FDA guidelines, the legislature for all of these things. And plus, as everybody knows, AI is a black box. So we have to make sure it's explainable, particularly for drug discovery. Every step is expensive and time-consuming. So you don't want to pour a lot of money, and later on, actually, it doesn't really make sense. And the data, everybody knows about it. Data quality, bias, ethical, social issues. Hallucination, I really want to mention about the hallucination. I mean, theoretical, like AlphaFold. So when I give them some structure for AlphaFold, it gives me some very weird predictions because it's as we in the modeling, right? So it always gives some prediction, always some result. Whether we call it false positive, false negatives. Whether it's accurate, nobody knows. And of course, AI can make up things. Actually, what I did last night, I did it on myself. I just said, tell me about myself. And they give me pretty good summary of myself. I really like them, maybe I should use them in the future. And then I asked them, give me more about AI, my expertise in AI. And they said, oh yeah, all of this. And then I found that he said that I was involved in natural language processing. I would say, oh, I didn't really do any natural language processing previously because I'm not an expert in this area. But because NLP part is part of the AI, so that's why he put it here. But now we are involved in large language model. Maybe I can say that now. It's small things. And then I further asked about my education. It says that I did a postdoc training sometime after graduation, but I never had a postdoc experience. That's why it didn't give me the where I was doing the right? It's interesting. This kind of things that it always gives you something. You have to identify what's the reason? Because the problem is because everybody in this area they have to have postdoc experience for their training. That's why for people doing drug discovery, AI drug discovery, you most probably know about NLP. So that's why I put it here. On the other hand, I think hallucination actually is a good thing in drug discovery
Shuxing,
because, okay, I will wrap up.
we got 5 minutes left, so I know we've got some questions.
Okay, yeah, I will finish it. So the basic idea is that I think hallucination on one side is bad, on the other side is good. It's really for drug discovery, it's really important because we need some crazy ideas. That's what I'm talking about. Crazy ideas in drug discovery. So we just apply this ALPA program. So basically, we'll build an AI platform, really predict the drug toxicities, enemy toxic profiles. Without in the future, we don't need to do any animal studies. This is also the actually the Congress passed this called FDA Modernization Act. Basically, is that you don't have to have animal studies in the future for FDA approval, as long as your data is really based on human biologies. And there is this conference, in the future, what will be like in the future. So we are thinking that people are thinking that actually in the future, the AI, because everything is about data, right? In the future, actually, the big tech company actually will run the AI, will run the clinical trials instead of big farmers. AI will control, will design everything. Maybe Theranos will be not a scandal anymore, will be personalized medicine will really come to the age. Drug repurposing, combination therapy, combination of AI, quantum computing, biocomputing. I think that's the future. So I think that's about my talk. This like cartoon was generated 10 years ago, so it's perfectly described my dream. So I want to build this thing.
Okay.
Okay, so I will open for questions.
That's great. Thank you so much for such an interesting talk. I know we had several questions online.